Hi everyone, this is Caroline with Fiveable, and we are in our 4th week of review. As you can see, this is just one of 3 live sessions left that are free for AP Bio. So if you're wondering how you can get access to more great content, you can do so by getting a Fiveable Plus membership, and the link is in there for you. Whoops, let me get this up. There we go. So the link's there for you. It's $35. Gives you access to all of our subject areas if you're in other AP classes. But most importantly, you can join us for our last 3 sessions, which are all going to be cram sessions. So it'll be the Friday, Saturday, and Sunday right before the AP exam on May 13th. And we'll be going over really explicit test-taking strategies. A little too late then. But test-taking strategies, final advice, my top 10 things to know for the AP exam, and all sorts of really, really important things. So that's only available if you're a Fiveable Plus member. Also with Fiveable Plus, you get all of my replays, which means that you have access to probably 30 hours of content at this point. Some fun weekend plans. You can just watch me talking at length. But you can go back all the way to the very beginning in September when we talked about evolution and the origin of life, all the way up through ecology, and then of course the other 3 review sessions that we've held thus far. So definitely check that out. The other 3 review sessions, we've covered a ton of content tonight. We are mainly focusing on heredity. That's going to talk a little bit about cell division, and then genetics, so Mendelian versus non-Mendelian genetics, and then body systems. So I'll try to get through as much as I possibly can. We will talk about the immune system, the nervous system, and the endocrine system if we can get to all of them. If not, we'll pick back up next week. As always, if you have questions, ask away, because this means a lot more if I'm answering your specific questions than if I'm just rambling to myself in my apartment. Now that sounds really sad. But no, truly, if you ask questions, I can make sure that you're getting the support that you need. Okay, so I'll be looking frequently to make sure that I'm answering those as we go. All right, let's jump in because time is of the essence and we only have an hour together and we have lots to talk about. Here we go. Okay, so we're going to start and picking back up because this is technically review, but we are going to start with cell division because that will lead us nicely into talking about heredity and genetics. Okay. Great. So with cell division, I think if you were back with me when I originally taught this, I have a great song to share with you about mitosis and meiosis, in case you forget the difference between them. And it goes a little something like this. Mitosis is in mitoses, meiosis is in meiosis. Mitosis is in mitoses, meiosis is in meiosis. It's a really great song. I feel like I'm going to become famous one day maybe because of it. But it's a really great way to remember the difference between mitosis and meiosis. So the song says mitosis is in mitoses, which helps you to remember that mitosis happens in body cells, also known as somatic cells. We start with a diploid cell, we end with a diploid cell. It is identical. We actually end with 2 diploid cells. What is a diploid cell? You can look on the right. It means you have 2 copies of each chromosome. And then meiosis is in meiosis, meaning ovaries. And so meiosis is used to create gametes or sex cells, egg and sperm. So that happens in the ovaries or testes, depending on if you are female or male. And then we start with a diploid cell, and we actually end up with a haploid cell that has half the amount of chromosomes as the body cells. So for humans, we have 46 chromosomes in all of our somatic cells. Our gametes, egg and sperm, only have 23, because those halves will come together with another half to make a whole, and that is the miracle of life. Seb, I see that you've already asked a great question. Is it important to understand the muscular system and specifics of the reproductive system and the parts? It is very much not important. In fact, you should spend 0 time studying those things. I guess I could backtrack slightly and say that with the reproductive system, if you're familiar with the hormones involved, that could be part of the endocrine system, but that's part of the endocrine system, not really the reproductive system. You do not need to know any muscles. You do not need to know any part of the reproductive system in order to pass the AP exam. The only systems that are covered are the immune system, the nervous system, and the endocrine system. So hopefully that helps answer that question and give you a lot less things to study. Okay, kind of going off of Seb's question, I'm already going on a tangent 5 minutes in, which is a good sign. With the AP exam, when it was redesigned in 2013, it was redesigned. And so if your teacher has been teaching for a really long time, they may like fall back into old habits of wanting you to know things like the muscular system or the reproductive system, because that used to be required. With the redesign in 2013, you have to do a lot less memorizing and a lot more applying. So that can be easier in some ways and a lot harder in some ways. Easier because you don't need to make like the 2,000 flashcards that I made when I took AP Biology. Harder in that you actually really have to understand what's going on so that you can apply it to new and different situations. So I'll talk about this a lot during like final review sessions, but the AP exam is designed to make you feel as though you know nothing. So if you've done a lot of practice problems with your teacher, which hopefully you have, sometimes you're like, I have truly no idea what these words are, and I have no idea what they mean by them. And no one knows what people do, but no other AP Bio student probably knows what those words are, and you usually don't need to know. So when we get closer to the exam, I'll do a lot of practice multiple choice just to prove to you that that's true and that it really is all about applying to new situations and not about memorizing all of these vocabulary words or body systems. Okay, side tangent over. Just took me a minute. Okay, so again, mitosis is in mitoses. So there you can see the overall end goals here and the purpose of these two. So mitosis is going to be happening when we are trying to make more cells. So perhaps you get a cut and we need to make some new skin cells, or your hair is replicating. I mean, your skin's always replicating because we're scraping off this external layer, and that's what makes up like 80% of dust, skin cells. So there are a lot of parts of your body that are continually going through mitosis, but it's also especially important when you're developing and growing and things like that. Meiosis is also happening, especially in males, because they're consistently making sperm for the entirety of their life. And that's, again, just happening in those two regions, either the testes or the ovaries, in order to create sex cells. Okay. Some key vocabulary, and again, I'm less concerned with you understanding every single word on this slide. I'm more concerned with you understanding the general purposes of these things. But just because when we talk about meiosis, we'll get into this a little bit. This is a chromosome. This is also a chromosome, but it's a duplicated chromosome. And we consider these two little arms that are attached in the middle by a centromere sister chromatids. Okay, so it's the same exact version of this chromosome. Okay, it's just duplicated. Okay, down here we have something a little bit different. These are homologous chromosomes. So each one of these, and sometimes students get confused because these straight lines don't look like the stereotypical chromosome that you have in your brain, which is an X. But these are both chromosomes, and they're homologous chromosomes, meaning they are different versions of the same chromosome. So we have 46 chromosomes in our body cells. We actually have 23 unique chromosomes, and we have 2 versions of each. We have 46 unique chromosomes. That's a misnomer. But we have 23 chromosomes, and we have 2 versions of each. So we have 2 versions of chromosome 1, one from mom, one from dad. Okay, so those chromosome 1s have the exact same genes on them in the exact same location, but we may have a different version of that gene, right? So like the gene for eye color, which is not this simple, right? But we may have one version of eye color from mom and one version of eye color from dad, but it both lives on the same chromosome in the same location. So those are homologous chromosomes. And that for some reason is some vocabulary that's sometimes tricky for students. Again, you don't need to be overly stressed about memorizing all the vocab, but it's important to kind of understand these distinctions in order to understand some of the nuances of mitosis and meiosis. Okay, so here's the process of mitosis. You do not need to know prophase, metaphase, anaphase, telophase. If you do, that's wonderful, but you do not need to. It is not required of you on the AP exam. There's an explicit note in the teacher's guide that says so you can trust me on that one. Okay, what you do need to know, though, is that we start with a diploid cell. So in this case, we have 2 chromosomes, and we end with 2 identical diploid cells with 2 chromosomes in each that are exactly the same as when we started. Okay, so that's going to be really helpful because you don't want to make a skin cell that's genetically unique. You just want to make a skin cell that looks like every other skin cell. Okay, so that's going to be our general process there. can see it again with our prophase, metaphase, anaphase, telophase. But again, you do not need to be familiar with those terms, but just the general process in and of itself with the chromosomes lining up, getting pulled apart, and then being genetically identical to the parent. See, we have a question. All right, Lillian, this is a great question. So Lillian's asking, when do mistakes in chromosomes happen, like the Down syndrome chromosome? So Down syndrome is caused by, it's actually caused in meiosis. So I can show you a little bit better when we get to the meiosis slide. It's caused by a non-disjunction, which means that the chromosomes, instead of separating, actually stay together. And it gives us an extra version of chromosome 21. So it's called trisomy 21. Which is what causes Down syndrome. So that happens in the mom when she's making eggs in meiosis. Okay, so that's what happens there with the separation of chromosomes. As far as mitosis, errors are very infrequent. I mean, they can certainly happen, but usually the cell then would stop dividing. And I'm going to show you an example in a second of how our cell knows to check for errors. So hopefully that helps answer that question as well. Okay, so the key takeaways here are that mitosis produces identical daughter cells. They have two cells from the parent cell, and that we replicate our DNA once in the beginning, in interphase. That's when DNA replication takes place, and then we divide once. Okay, so we're doubling and then halving, so we end up with the same as what we started. So here's that cell cycle regulation that I was just referring to. So it is important to know that this is regulated so that we don't have those errors that Lillian was talking about. So we start with G1 phase, and in G1 phase, we're going to be replicating a lot of our organelles as well as the actual like cytoplasm so that we have enough to make two cells. So at the first checkpoint, G1, we're going to check for growth factors and nutrients in the cell to make sure that both cells have the appropriate amount of nutrients and any damage in DNA to start. We haven't synthesized the DNA. You can see that's coming up next in S phase. But just any DNA damage that may have been pre-existing. Things like UV rays and chemicals and different things like that can cause damage into our DNA. So G1 checkpoint is basically like, hey, it's like the checkpoint when you're writing a paper where your teacher's like, let's check in to make sure that this isn't trash. Because if it is, you shouldn't keep writing it, right? Because you're going to waste a lot of time writing a bad paper. So they're going to kind of have a checkpoint. They'll read through what you have so far and see if it's any good. If it is, you can keep going. If it's not, you got to stop and rewrite. Okay, so that's what happens here. And this is a big checkpoint, this first one, to see if the cell is ready to divide. And if it's not, it goes into resting state and it does not divide. Problems with these checkpoints are what leads to tumors and things like that. Okay, so it's important to know because a lot of times they'll put questions in about what can go wrong in this cell cycle regulation and how that might lead to a tumor. Okay, so after the G1 checkpoint, we're cruising through and we go through all of our DNA replication. So DNA is replicated. We talked about how that process occurs last week if you were tuning in. And then we go into G2 phase. And in G2 phase, this is where we're going to replicate all of our organelles and all the final things that we need to replicate in order to make sure that we can form two cells out of one. G2 checkpoint then checks for cell size and DNA replication. As you can see, DNA replication is a big one. If we've made an error in our DNA synthesis, we want to make sure that that doesn't get passed on. And so the cell, if there is error, will actually just stop replicating, and therefore we will not have that mutation in our or in the new cell that is formed. Okay, if we can make it past the G2 checkpoint, then we go into M phase, which is metaphase, or not metaphase, which is mitosis. So it's actually a relatively short process, as you can see. It takes up much less time than everything else, even though we talk about it the most. What happens here is we have one final checkpoint, which is the metaphase checkpoint, which checks for chromosome spindle attachment. So this is where we could have problems in meiosis that lead to Down syndrome. And if you can see here, if these are not properly attached, let me get my little pointer out. If these are not properly attached and they're only attached to one side of the cell as opposed to both, then they're not going to pull apart correctly and we're not going to have equal numbers in our daughter cells. So that's actually a really, really, really important part of making sure that we end up with genetically identical daughter cells, or at least daughter cells that have the right amount of chromosomes in them if this is in meiosis, because this will happen in both meiosis and mitosis. Sorry. Okay. Ubiquitin. I see someone just asked a question. Yes, Aditi. Hello. Welcome back. You've been here before. Ubiquitin is a protein that is going to target, it's going to be involved. Let me get back to my slides. Sorry. It's going to be involved in checking the DNA replication. So it usually checks for like thymine dimers or other sort of, what's the word I'm looking for? I swear, review season is really tough. I have office hours every day during review season. So I'm just like talking constantly if I feel like my brain is in a different place right now. But it's going to check to make sure that our DNA is linked correctly and that we don't have any sort of connected base pairs. So thymine dimers sometimes form, which is where two thymine bases actually connect, and it can create a lump in our DNA, which would be a mutation. So ubiquitin is going to come and check for errors in replication like that. So that was a great question. Thanks for asking. Okay, perfect. Okay, so meiosis is going to be a little bit different then. So there's definitely similarities. We are going to duplicate our DNA. And as you can see here, so we're starting with 4 chromosomes, but we have 2 versions of the red chromosome and 2 versions of the blue chromosome. Or although I guess the way that they divided this, no, that makes sense. Yes, okay. And so when these homologous chromosomes are lining up, I guess they did big and big and little and little. Sorry, I'm looking at how this diagram is set up. When these homologous chromosomes pair up with one another, so these would be the same version of like 2 big chromosomes, 2 little chromosomes, we have this process of crossing over, which I'm going to show you in a second. But it is one of the key ways that we get genetic variation in our offspring. So it's really, really important to know about. Right? And in crossing over, exactly what it sounds like. Part of one version of our chromosome transfers with another version of our chromosome. Now remember, these are technically the same chromosomes. So if these were both chromosome 1, we're exchanging genetic material at the same exact location. So the genes are still going to be transferred. We're not going to lose a gene, right? We're not going to gain an extra gene for eye color or lose a gene for eye color. We're going to transfer the gene for eye color. So now there might be a different version, like dad's version of eye color is now on mom's version of that chromosome, and mom's version of eye color is now on dad's version of that chromosome, which is going to lead to a lot more possibilities than just what we see when we do like a Punnett square and we're wondering what the inheritance patterns might be, okay? So another thing with meiosis, we're duplicating our DNA once, but then we're dividing twice, which is why we're ending up with these haploid cells that are all genetically unique from one another. Okay, so another thing that's involved in here is something called independent assortment, which I have a whole slide on as well. Okay, and that's what tells us that these chromosomes are not tied to one another. Okay, so the version from mom for chromosome 1 and the version from mom for chromosome 2 don't necessarily have to be together. And in fact, in this instance, they're not. They're actually separated. And the version from mom is paired with the version of chromosome 2 from dad and vice versa. Okay? But those abilities of those things to switch and independently assort from one another is what gives us so much variation in the egg and sperm possibilities. In fact, it leads to like 1 in 8 million chance of you being you, just from the two sets of DNA that exist. Sorry, I'm like trying to bounce back and forth and also answer your question. Ooh, okay. Sorry, I was just looking at your question of chiasmata, and I was trying to see if I can show you on this. So the chiasmata is going to be the area in which we're crossing over. Okay, so trying to show you on this. It's kind of hard to see on this diagram, but like wherever these 2 chromosomes lined up and crossed over and exchanged this DNA would be that point of chiasmata. Hopefully that answers your question. don't need to know that vocabulary word for the AP exam. I also, with the ubiquitin question, it's very helpful as well, but they are always going to give you the context of what ubiquitin does, and you just need to apply it to that situation. So don't feel like you need to pull out the word ubiquitin or chiasmata or even homologous chromosome out of nowhere. They're always going to provide you the context, and you just need to be familiar with the overarching ideas. So hopefully that gives you a little bit of peace of mind. I don't know. I've looked at so many AP questions in my years of teaching this class that I can tell you with certainty since 2013, you do not need to know anything like that. Have you guys done, I guess I can't really ask 33 people this virtually, but hopefully you've done some practice exams with your teachers. Can a few people just respond? I'll talk about the law of segregation with independent assortment. Okay, seems like most people have. I'm glad to hear. Oh, we got a mix. A lot of no's as well. Okay. That's fine. Hopefully you've at least done practice questions. I'm seeing so many answers now. Well, hopefully you've at least done some practice questions. So what this is telling me is that maybe we need to spend a lot more time doing practice questions. Because I want to kind of prove to you definitively that some of this vocab that you're stressed about, you don't need to be stressed about, because it's not what the exam is going to focus on. Okay, so Anusha, to go with your question, which is a very good one, you do not need to know PMAT, like prophase, metaphase, anaphase, telophase for meiosis either, or really the differences between meiosis 1 and 2. What I'm more concerned with you knowing is that there are homologous chromosomes in meiosis, right? And those homologous chromosomes are going to line up and be capable of crossing over, and then they're going to divide from one another, and then they're going to divide again. So if you understand crossing over, independent assortment, and the fact that homologous chromosomes are being separated and that leads to variation, then that's the important thing to know. It's much less important that you know the specific differences between the two. But it's good to know that homologous chromosomes separate and then sister chromatids separate in meiosis 2. Okay. Sophia, I see you asked a question all about DNA fingerprinting, electrophoresis, genetics, heredity. I'm going to table that until we get a little bit into that topic, and then I'll come back. So I promise I will answer it, just not right at this moment. Okay. So meiosis, the key takeaways that you need to know are that they produce genetically unique daughter cells. They produce 4 cells from the parent cell. We replicate once and divide twice, which is why we end up with haploid cells that have half the amount of DNA. So in an egg or a sperm, there's one version of chromosome 1, one version of chromosome 2, all the way up to 23, as opposed to in our body cells where we have 2 versions of each. Okay? All right, so let's talk about some of those important things then. So independent assortment, which is going to be the same as the law of segregation, whoever asked that question in the chat stream. Independent assortment is just this. So there's a couple different possibilities here. So here, this is actually the same kind of setup as we saw in this example, where we have 2 big chromosomes, a blue one and a red one. They use that to represent mom and dad. And then we have 2 small chromosomes. So we'll call the big ones chromosome 1 and the small ones below it chromosome 2. Okay? And when these homologous chromosomes are lining up at the mitotic plate, or meiotic plate, I should say, because this is meiosis, they can line up in any which way. So you can see on the left and the right, these are the only ways in this circumstance, because we only have 2 chromosomes, that we can set these chromosomes up at the meiotic plate. So on the left-hand side, it just so happens that dad's version of 1 and 2 end up on one side, and mom's version of 1 and 2 end up on the other. So when we separate these out and finally divide twice, you'll see that we have a combination 1, which is dad's version of both chromosomes, and combination 2, which is mom's version of both chromosomes. On the right-hand side, it is just as likely that we'll have dads on the left and moms on the left, and moms on the right and dads on the right. So now we have kind of a mix for chromosome 1 and 2 between mom and dad. So when we divide, you'll see combination 3 has dad's version of chromosome 1 and mom's version of chromosome 2, and combination 4 has mom's version of chromosome 1 and dad's version of chromosome 2. So there are 4 different daughter cells, 4 different egg or sperm, depending, that can be created because we have 2 different chromosomes here. So it's 2 to the 2nd possibilities. So with us, we have 23. So if you do 2 to the 23rd, the exact number off the top of my head. Let me pull up my handy dandy calculator on my computer. Although I don't know if it does exponents like that. It definitely doesn't. Anyway, I think it's 8 million different possibilities of egg and sperm that can legitimately be formed just based on this principle. And that has nothing to do with anything else that we're talking about. Okay, so that's pretty crazy. It's important to know about for sure. You also do this all the time when you're dividing the alleles of like a heterozygote when you're doing a Punnett square. Okay, so when you're like big A, little a, and they each get their own column, that's independent assortment. You're saying there's a 50% chance they're going to inherit this one, 50% chance we're going to inherit the next one. Okay, I see we have a few questions. Let me pause really quick. Okay, so Cam, to answer your question, there will be nothing more covered on the AP exam than evolution. The reason is evolution. Oh, thank you, Logan, for doing the math. 8.3 million, and Anusha. You might count on you guys. You're the best. There's nothing that will be covered more on the AP exam than evolution. Okay, it is an entire big idea in and of itself. So if you guys are familiar with the 4 big ideas, big idea 1 is solely evolution. Okay, so there's something to be said for that, right? It means that it's a quarter of the important things that you're learning about, even though it's just 1 unit. The other thing that's really, really important about evolution is that it can truly, it ties everything together, right? So they can ask you a cell question, or they can ask you a cellular respiration question, but they're asking you about what is the most evolutionarily conserved pathway. So they're tying evolution and cellular respiration to get to the answer of glycolysis. So that's going to be the most heavily hit thing on the exam, pretty much without fail. As far as genetics versus ecology, kind of your guess is as good as mine. It really depends on the year. So every year after the exam, my kids have different opinions on what was covered. And when I look at the free responses, I never get to see the multiple choice. When I look at the free responses, there's always like kind of a heavy topic. So ecology happened recently. Cells actually happened pretty recently. It just depends on the year. If I knew exactly what you were going to be tested on, all of you would pass and all of my kids would pass and life would be so good. But it's kind of a crapshoot. So we can use history to help us, right? But that's kind of what I can tell you. Ecology, the great thing about ecology is half the time it's a graph or a figure. And if you are a logical person, you can answer that question without knowing anything about the words they're using. Okay? But part of that is just having confidence in yourself. And that's why we'll start practicing. Maybe next week we'll just start like fully practicing some multiple choice and things like that so that you guys can start to feel a little bit more confident in that. Plant structure and anatomy not covered. The anatomy of a plant is not covered at all. The only real thing that's covered with plants is a little bit on abiotic signaling, so like phototropism and that famous experiment, which you've probably seen, where they have the different plant shoots that are phototroping, if that's a verb, towards the light, and they have like a cap on one and they cut one off and things like that. That shows up because they really like that diagram. Once they buy a diagram, they like to use it multiple times. Fun fact. And photosynthesis comes up all the time. But other than that, plants are essentially not touched. Okay, and again, Sophia, I promise I'm coming to your question once we get into genetics and heredity a little bit. Okay, back to where we were, crossing over, super straightforward. If no one has questions on this, I'll kind of move on. But you can see, like, Dad has all dominant versions because this is a duplicated DNA, so that's why we have the same. They couldn't be heterozygous at this point because this is just duplicated blue DNA and red DNA. But then they cross over, and now we have the lowercase version of this gene and the uppercase version of this gene here. So after independent assortment, there's going to be even more possibilities for variation. Okay, lastly is random fertilization, and this basically just refers to the fact that you don't know which egg is going to fertilize which sperm. And so, you don't know which one Dad is giving and which one Mom is giving, and that's going to provide some semblance of genetic variation as well. I really like to focus a lot of time on these big 3 things because what you need to know is that genetic variation is key to life. It's the key to natural selection. We wouldn't have natural selection if we didn't have genetic variation, because no one would have a better chance of surviving than anyone else. Okay, so you can't be more fit if everyone's the same. So genetic variation is truly the key to us evolving. It also keeps us hardy and makes sure that we're not being, susceptible to disease or being wiped out. If you've heard about like some things, like all of our bananas are genetically identical that we sell in our grocery store. And so if something happens and there's like a virus or a plague or something that wipes out that strain of banana, we're done for because there's no variation in the population. They're all genetically the same. So that's why I like to focus on these three, because a lot of times you can get the right answer if you just talk about variation or something that has to do with natural selection, right? Anything like that will help you for sure. Okay. Law of segregation and law of dependent assortment are the same. Hopefully that answers your question, David. Okay, let's see, more questions. What about, start answering, Anika and Sophia, I promise I'm still coming to you. What about human physiology is needed for the exam? In my study book, it is a very large chapter, but it says not all of it is needed. So Anika, if you have the Cliff's Notes study book by any chance, that's the one that I use in my class. And for physiology, the only thing you need to know is the nervous system, immune system, and endocrine system. They may ask a question that involves a different system, but they would provide you all the necessary information that you need to answer it. So the only things that you need to be familiar with are immune, nervous, and endocrine system. So usually in review books, like in the review book we use, they talk about the circulatory system, the respiratory system, all of those. Don't need to know it. Okay. And we're cruising. Let's get into genetics briefly, and then I can answer Sophia's question. But honestly, genetics hopefully is one of the easier things that we talk about because it's usually covered in like basic biology classes. But definitely ask me questions if you have them. Okay, so just a little background. There's Gregor Mendel himself, the father of modern day genetics. The only reason I have this picture up is because you need to know about on the right where you have P generations, F1 generations, and F2 generations. Those terms will be thrown around in free response or multiple choice questions, and you need to be familiar with what they mean. So the P generation is the parent generation. Oftentimes they are true breeding parents if this is a test cross, which in this case it is. Then you have the F1 generation, which is going to be all hybrids. You see that they're all purple. That tells you right off the bat, without having to be told, that purple is dominant. Oftentimes they will have trick questions like this for you, where they say, we have a true breeding purple flower and a true breeding white flower, and all the offspring are purple. You have to be like, okay, that means that purple is dominant, even though they didn't tell me that, okay? Then the F2 generation is a cross between two F1 generations. So that's important to know too, because sometimes they'll just say in the F2 generation, but they didn't explicitly tell you to cross the parental generation all the way up in the beginning, okay? So this is a classic example of a 3-1 split then, because we have heterozygotes for our F1 generation, since we have a true breeding purple and a true breeding white flower. Those are all heterozygous purple flowers, which means that approximately 3/4 will be dominant, 50% of which are carriers or heterozygotes, 25% of which are true breeding, and then the other 25% are the true breeding homozygous recessive white flowers. So hopefully that this is all stuff that is ringing bells. Okay, cool. Just a few other vocabulary situations then. A character is going to be what we call like seed color, seed shape, pod shape, that kind of overarching description. And then these guys are going to be the traits. It's either purple or it's white, right? That's the dominant trait or the recessive trait. So purple is the dominant trait for flower color, which is the character, okay? So just like a couple other kind of key vocabulary there. But again, basic Mendelian genetics should be the least of our problems. After Mendel is when DNA was discovered. So he actually did not know about DNA, which is absolutely nuts. We can't fathom that. A lot of back in the day, they thought that proteins were what we inherited, not DNA. So that's kind of interesting. But there's also this term allele that you need to be familiar with. And the allele is just a version of a gene. So it codes for a trait. So we have the allele for purple flowers versus the allele for white flowers. This individual having both would be heterozygous for its genotype. okay? Again, other basic vocabulary: homozygous dominant, heterozygous, homozygous recessive, phenotype versus genotype. I'll just keep kind of zooming through this as long as nobody has any questions. Emily, don't waste your time memorizing any scientists. Absolutely not necessary. They would explain to you. Actually, the only, I take that back as I say this, the only scientists that I would be familiar with are Miller and Urey and their experiment for early Earth, the origin of life. So Miller and Urey and their origin of life experiment is really the only one that you need to know. Drexy, great question with chi-square. I do not have the software that I need to show you a chi-square tonight, so we'll have to postpone that. I was going to do an entire math review closer to the exam, and chi-square will definitely be one of them, and I can explain that in detail to you. What I will say is that a chi-square hasn't shown up relatively recently, which is not necessarily a good sign for you guys. It means that it might be coming, but it's basically like an all or nothing situation. The past few years, there literally have been zero chi-squares. So again, that could mean that it's coming this year, but it also means that you can get away without having to know it at all. But we can certainly go through it because it's actually not too bad, and the formulas are given to you. You just need to know, and the math is very easy. You just need to know what your answer is telling you. So we can certainly go through that. If we have time at the end of today, I can try to pull up that program. Otherwise, I can't. I have to write, and if I literally write and then flip it and show you, it's a mirror image, and so you can't see anything that I'm saying. So I can't do it without writing things down for you. Kelly, for gene mapping, no, I want to say tentatively no. If anything, it might be one problem. I would say with gene mapping, the only thing you need to know is the more time that something recombines, the further away those genes are. And if it recombines 50% or more of the time, then they're on different chromosomes because that's just independent assortment. Yeah, so I would just know that the larger the amount of recombination, the further those genes are from each other, but you're not going to have to map out a gene on a free response or anything like that. Hopefully that answers that question. Milka, why were proteins considered genetic material? Proteins are genetic material. I mean, they're not like inherited genetic material, but the reason, I mean, it contains your genetic material because it contains, we use it for inheritance all the time and figuring out how closely related things are. Because we can look at your amino acids, and that tells us a lot about your DNA, which is your genetic material. Why were they considered the inherited genetic material? Because there were 20 amino acids and only 4 nucleotides, right? So we have 20 different amino acids, and then we only have adenine, thymine, guanine, and cytosine. So they're like, there's no way that the key to life can be held with just 4 bases. It has to be this thing that has 20 bases. Okay, Sophia, I'm just going to answer your question now because you've been waiting so patiently. Okay, so fingerprinting, you don't need to know anything about. Gel electrophoresis, DNA fingerprinting, yes, that's a different story. That you might need to know a little bit about. So it's kind of involved in a lot of different things. So with the importance of DNA in and of itself, and then gel electrophoresis. So things that I can tell you, let me pull up a PowerPoint actually really quick. And I'll try to talk while I'm pulling this up to try to multitask and make sure we're using all of our time wisely. With gel electrophoresis, all that does is allows us to, with anything biotechnology, it allows us to visualize DNA. Right? We can't see DNA. Even if you get a big old clump of DNA, and if you've done a DNA extraction lab, maybe you've seen DNA, we can't understand what it's coding for there. Now we have really, really, really amazing technology that allows us to truly dive deep into DNA and actually, come up with we can have our whole genome expressed in, a couple days because everything has become so quick and so informed. But as far as gel electrophoresis goes, this is just another way for us to visualize what DNA is showing us. So what happens is we use something called restriction enzymes, and restriction enzymes cut DNA at one exact specific location every single time, the same specific location. And what that does for us is it allows us to determine how long different DNA segments are. And if DNA segments are the same length, then they probably have the same bases in them. So I really need to pull this slide up. This will make a lot more sense of what it's saying. All right, bear with me. Okay. Of course, it's going to take forever to load because I really want to show you on a visual. So I guess while this is loading, I can explain the actual structure of a gel. So gel has little micropores in it, okay, that DNA can move through. And the other thing that we do, I used to run a bajillion of these in college. Here we go. So we have our solution, which we've mixed with restriction enzymes, and then we load it into this gel, and this gel has specific chambers, and then we put charge on it. So we have a positive charge at this end and a negative charge at this end. DNA is going to be negative charged, okay? We also put it in a dye that is going to enhance its negative charge. So the negative charged DNA is going to be pulled through the gel, and you can kind of think of it as like one of those like Plinko boards where the ball is like bouncing through and hitting the little pegs as it goes down. That's basically what's happening with DNA. It's getting pulled through towards this positive charge that it's attracted to. And then we have this pattern, this banding pattern is what it's called in the end. And so you really don't need to know all those specifics. What you need to know is what it kind of means for us, okay? So if you can think about cutting DNA up in chunks, right? If all of our DNA is being cut up in the same with the same bases. Let me see. Here's restriction enzymes. So this is how restriction enzymes cut. They'll cut at the same region, and then we have these protruding ends. So we'll use the same restriction enzyme on all of our DNA and then run it through a gel. And if you're related to someone, you should have very similar chunks to them. So this is how it's involved with whatever you were saying about DNA fingerprinting. So what you can see here is that we have a paternity test, which is always very exciting. So we have a mother, a child, and 2 possible fathers. And so the child's banding pattern has these 6 segments that are significantly similar to the mom, and also segments that are significantly similar to male number 1. And so we can use it for things like paternity in order to determine that kind of stuff. So do you need to know all the logistics behind it? Not really. I would say biotech, which is what you're kind of referring to here, is actually a relatively small portion of the overall AP exam. But it is kind of important. You may have to analyze a diagram like this and be able to tell that there are DNA similarities because of restriction enzymes. And so if you are genetically similar to an individual, you're going to have a similar banding pattern. I hope that answers your question. If it doesn't, feel free to ask another because you've been waiting so patiently. But that's kind of the short answer, I guess, to your question. But I wouldn't stress too much about knowing like all of the logistics. You don't need to know about the positive charge. I'm just explaining that to you so that you understand. The only other thing is that, oops, wrong PowerPoint. These segments down here are going to be our shortest segments of DNA, and these segments up here are going to be our longest segments of DNA because shorter things are just going to be able to travel through the gel much more quickly than longer and bulkier things. The further the DNA base travels, the smaller it is, right? Because if you think about like a thing full of pores, right? Smaller DNA segments are going to be able to make it through much quickly. They're going to bounce through much more quickly than if you're trying to send like a beach ball through, right? Which would be larger segments of DNA, which are going to remain up at the top, right where you loaded them. Seb answered your question perfectly. You don't need to know all of the specifics behind them. I wouldn't like waste a ton of time memorizing. If they show up, they usually tell you what they discovered. So it's really not worth your brain space. There are definitely more important things to know. So heredity directs, I'm telling you right now, is just like the basics of like Punnett squares and inheritance. But if you understand independent assortment, crossing over and random fertilization, especially independent assortment, you can get through the vast majority of these things. that you would see on an AP exam. I guess another good thing to know is like autosomal versus sex-linked traits that are inherited. So sex-linked is going to be something that's carried on the X chromosome, so it's going to be more prevalent in males than in females because they only get one version of X. So if it's the version that has whatever disorder, like color blindness on it, it's going to be a whole lot more prevalent. But if it's autosomal, we're going to have an equal distribution between males and females. That's a good thing to know. Pedigrees comes up on occasion with heredity, but not super frequently. I just saw one kind of recently. But again, if you understand how things are inherited, then you shouldn't have to like spend a whole ton of time memorizing pedigrees, if that makes sense. For DNA replication, I did a whole thing on it last week, so you can rewatch that replay. But you should be relatively familiar with the main enzymes, so like DNA polymerase, helicase, primase, ligase, and the overall nature of DNA replication, which leads to leading and lagging strands based on the anti-parallel nature of DNA, and how one end runs from 3' to 5' and one end runs from 5' to 3'. Okay. I'm going to switch back to my original PowerPoint then. We haven't gotten through genetics yet, but that's okay. I think we can zoom through it really quickly, and I'm actually much more happy to answer your questions than to just cruise through, PowerPoint slides. That's way less exciting. And I never teach with PowerPoint slides, so it's weird for me. Like in my class, I never lecture, so it's weird for me to have these PowerPoint slides with you guys. Okay. All right. Hopefully we're good with all the genetics basic vocabulary. Okay, so very briefly then I'll run you through non-Mendelian genetics because sometimes these problems pop up. I just saw one today on a practice test that I was doing with my students. The reality is that we've all learned about Mendel and Mendelian genetics for forever, but the vast majority of traits are not inherited this way. Okay, so everything we've looked at so far have two alleles, dominant and recessive, with predictable inheritance patterns. But there's so much more, like codominance, incomplete dominance, multiple alleles, and then sex-linked traits, which I just referred to briefly. So with codominance, you want to think co meaning together or both, like a co-captain. So if alleles are codominant, then both alleles are dominant. One is not more dominant than the other. Okay, so that means the heterozygote of this type of inheritance pattern is going to have both dominant traits showing. So the perfect example is the checkered chicken. We have a black chicken and a white chicken of a certain breed. And when they breed, they mix together. So we'd have like two black alleles, two white alleles. The offspring gets a black and a white allele, and we have a checkered chicken. They're showing both traits. With incomplete dominance, neither allele is dominant. So instead of seeing both alleles show up, or both traits show up in the offspring of a heterozygote, we see neither trait showing up in the heterozygote. So if we have a red flower and a white flower crossing and it is incompletely dominant, then we would have some sort of intermediate, in this case, pink. So it's not red with white spots. It's not like red and white are both showing up. There's just this like blend. So really technically neither of them are showing up. And then the last one that we'll kind of touch on briefly, and then I'll probably just try to answer some more of your questions for the last 10 minutes because I don't really want to get into the immune system with just 10 minutes left. So multiple alleles, many genes exist in multiple forms. The common example here is going to be blood typing. So what you can see here are the 8 blood types that exist in humans. Okay. And what you need to know is that we have 2 codominant alleles, A and B. One is not more dominant than the other, hence blood type AB. And we also have 1 recessive allele, and that's going to be blood type O. So if you're blood type O, the only reason or the only way that you would have that blood type is if you have 2 recessive O alleles. Okay? What you can see here is that if you have a B allele and an O allele, or an A allele and an O allele, that A or B is going to be dominant, and you have type A or type B blood. Okay? It's only if you have 2 recessive alleles that you would have that type O blood. And again, A and B are codominant, and you can see that here because A plus B equals AB blood type, which exists. Yeah, so that's just kind of like an interesting, we have some like a couple different things that are at play there. Then there's also pluses and minuses, which is a different protein factor, so we won't get into that. Okay. So I can jump into the immune system unless people have questions. I have done cell structure. Unfortunately, that was in review session 2, but you can watch the replay. The good news with cell structure is the really big thing that you need to know about the structure of is the plasma membrane, okay? And then you just need to be relatively familiar with like the Golgi, ribosomes, nucleus, mitochondria, lysosomes, vesicles, things like that, but more how they relate to protein synthesis, I would say. Most questions that I see ask students to be able to map out protein synthesis. There was a free response last year that asked students to do this, and students needed to know that protein synthesis occurs in the rough endoplasmic reticulum, then it moves to the Golgi, then it moves to the vesicles, and then it moves outside of the cell or to another part of the cell. So as long as you know that the general purposes of those guys, you should be pretty good, and you need to be heavily familiar with the plasma membrane. Any other questions that I can answer for you in these last 10 minutes? Otherwise, I can certainly start the immune system. We'll receive questions regarding non-specific immunity. Zachary, ooh, where did that just go? Did you delete it? Did I delete it? What just happened? Well, Zachary, I'll answer your question anyway. Non-specific immune response you definitely should know about. So I can show you really briefly on these slides. You should be familiar with this section, right? You need to know that the first line of defense, you need to know that non-specific immunity is what you're born with. You need to know it's the first thing to act. It's faster and it's non-specific, meaning it doesn't have memory. So it will just attack anything that's not you. Those are the big overarching concepts that you should know. And then I would know a few examples of each. So that like first line of defense might be skin or mucus. The second line of defense might be a fever or an inflammatory response. But that all of those things are just going to target anything that's not you. It's a lot quicker. You're born with it, but it also doesn't have memory. So it's not going to remember the next time it sees that thing that it's fought it off before. So you should be relatively familiar with that. Also, I like students to be relatively familiar with passive immunity. Passive immunity is the immunity that you acquire when you're developing in your mother's womb and like right afterwards. So passive immunity can be some of this specific stuff like antibodies, but it's antibodies that are transferred from mom to baby through the placenta or through breast milk during breastfeeding. So a lot of antibodies are actually transferred that way. So it's still this kind of specific defense, but it's not because you got sick with something. It's because your mom gifted you those antibodies. Drex, great question. So vaccine is technically going to be, well, it's kind of a mix, but it's technically going to be active because you're not getting sick, but let me show you. I think I have a slide. Please, what did I do with it? Here we go. So vaccines have to do with your B cells and antibodies. So what you can see here is that the secondary response that the B cell has to a specific antigen, which is going to be what the kind of red flag on the outside of a pathogen is infinitely stronger. So if this is 10 to the first, this is 10 to the fourth. So a heck of a lot stronger and also a lot faster. You'll see that the time that it takes to kind of escalate this response is a lot quicker than here, where we're first exposed to the antigen, and it actually takes almost 7 days for us to even start making antibodies, whereas here it's taking us 7 days to get to the peak of antibody production. And so that's what a vaccine utilizes. So when you get vaccinated, okay, you're tricking your body into having this first response. It's the first exposure. But the vaccine is going to be a weakened or heat-killed virus. Okay, so that's going to be injected into you. Your body starts to recognize the antigens on the surface of that virus, and it starts to make a small amount of antibodies, but you're not getting sick because the virus is heat-killed or weakened. Okay. Then the second time you're exposed to it, like in real life, we have this full-fledged second attack, and you don't even know that this virus even entered your body. So it's really pretty crazy. So you could have been exposed to tons of different viruses in your life and never even known it because your body has this crazy secondary response. Any other questions? No, that would not be passive immunity. Passive immunity is really just mom to child, okay? So because you got bit by the snake, you're exposed to that thing in real life. Okay? Passive immunity is like you haven't been exposed to that thing, you're just getting the antibodies to it. Whereas active immunity is you are being exposed to that thing and your own body is having a reaction to it. Okay. Oh, homology, introns, and exons. Okay, perfect. I can answer this for you, Zachary. Introns and exons I talked about last week. So definitely go back and relook because I won't spend a ton of time talking about them. So homology, I don't know in what context you're referring to. If it's homologous chromosomes, it just means it's the same chromosome. We might just have different versions of them, like the version from mom and the version from dad. Homology just means same. Homo means same. Okay, so homologous structures are structures, evolutionarily speaking, that are the same but have a different purpose now that have evolved from a common ancestor, like the hand of a human with the paw of a cat with the fin of a whale. Introns and exons has to do with transcription. I'll just give a very brief summary. So transcription in eukaryotes, we make pre-mRNA, which is not something that you probably learned in the past. You just heard that we made RNA, or mRNA, sorry, but pre-mRNA then gets processed further only in eukaryotes, okay? And what happens is we take that, we add a 5' cap that guides us to the ribosome and a 3' poly A tail that protects us from enzymes called exonucleases that are trying to snip away at this single-stranded mRNA. And then we have these introns and exons. So we have something called splicing. We have a spliceosome that comes through and removes introns. Okay, so I always say introns stay in the nucleus, exons exit the nucleus. They're actually part of the message, the mRNA. Someone last week said exons are exciting people, so you keep them around. So that works too. But regardless, we're removing the introns. They do not go and get turned into protein, and the exons get stuck together, and then that is the message that actually leaves the nucleus. The cool thing is an intron is not always an intron, and an exon is not always an exon. So the next time around, we could remove a different section of the mRNA, and therefore we'd actually be making a slightly different protein, even though the original gene was the same. Seb, MHC1 and MHC2 are classes of, so it's, let me try to get a picture. Well, actually I might have one in this slide set. I guess the good news is you don't need to know about them. So you can see it here, high levels of MHC class 2. It's just these little projections on the surface of cells that mark either yourself or not yourself. Okay, and so that's what those molecules do. But you don't need to know the differences between them. You don't even really need to know that they exist, but that's what they do. It's just the proteins on the surface of your cell that are actually being identified as you. Okay, so we have this self marker called MHC. So when your T cells and your B cells are maturing, so your T cells mature in the thymus, hence the name T cells, and your B cells mature in the bone marrow, hence the name B cells, they're learning to recognize your own MHC. So they're learning to recognize what makes you you, which is why if you get an organ transplant, your body can reject it very easily because that organ is covered in a whole other protein, a whole other MHC. Any other questions? Okay. I'm trying to think if there's anything I can cover in the last 2 minutes so we don't waste any time. I can just show you this very last kind of adaptive immune response overview. So you can just take a look and I can leave you with this. So first line of defense and second line of defense we talked about briefly. With the adaptive, acquired, specific immune response, whatever you want to call it, it's all the same thing. This is where we actually have memory and remember things. This is what involves our B cells and our T cells. So on the left-hand side, you can see the B cell response. So B cells are always going to attack free antigens in the blood, so free pathogens that are floating around, right? This is usually what happens first before your cells actually get infected, right? We just have these pathogens floating around. So this leads to the humoral response, and we say humoral because the humors of the body are the fluids of the body. So the B cells are going to take care of these. They recognize, they usually eat eat up that thing because they are antigen presenting cells themselves. And then they will produce two kind of separate kinds of B cells, plasma cells and memory cells. Memory cells are going to remember that thing for next time, and plasma cells are going to make a crap ton of antibodies, which can then trap the pathogens that are in the blood. Okay. On the right-hand side, if we have the cellular response, which is antigens actually infecting cells, pathogens, antigens actually infecting cells, that's where T cells come into play. Okay, so T cells, specifically on the right-hand side, that would be cytotoxic T cells or killer T cells, will actually go and kill the cells that are infected because we can no longer have them in the body. So we have the cytotoxic T cells and then memory T cells, which we'll remember for next time. Then in the middle, we have these amazing things called helper T cells, and helper T cells are able to recognize antigens and then send off an alarm of signaling molecules in order to let the T cells and the B cells know what we're up against and that we need backup. So that's my oversimplified crash course. We'll pick back up with the immune system, and then we'll spend a lot of time on nervous system and endocrine system next week. I will also make sure that we spend at least half the time doing practice problems, practice multiple choice, so that I can walk you through my own strategies that I talk to my students about all the time. The bottom line is you need to see these questions to see what you're up against because they're really, really, really hard. Okay, so definitely tune in next week. It's honestly essential that you're practicing these questions if you want to get that 3, 4, or 5. I know we were all over the place tonight, but I'm okay with that because I'd rather kind of fumble through and make sure that I answer all of your questions as opposed to just cruising through the things that I have planned out. So come with your questions. If you're studying over the weekend, which you should be, you definitely should be studying over the weekend, write down a bunch of questions and we can spend a lot of time next week answering them. As well as doing those practice problems, okay? Because that's what's going to be most beneficial to you actually passing this thing. It is a beast, but you are fully capable, okay? So think of those questions, and I look forward to seeing you guys next week. Have a great rest of your week, and good luck studying.